Tres, dos, uno. Hello, everybody. Welcome to this podcast, Just Gente. Today we have a special guest, an old friend of the podcast. But this podcast is going to be different because this is going to be in English.
It's
As
gonna be
Rick
hard.
said,
Thank you
so
so much for the invitation, man. And
PH Héctor Pool. So you are chemist, master in science and technology, food science and technology, and you have your PhD in food science and technology as well. Uh, right now, you are a teacher in Universidad Autónoma de Querétaro, and your research field is based on nanotechnology, right? So,
right.
welcome to the podcast. Let me give you applause.
Thank you, bro, for the invitation
Yeah,
again.
this is
And
gonna be different because I always have what like the what I already know what to say at the beginning of the podcast. And yesterday I did another one in English and was like, I don't have an intro for this. It's like I should write an intro in English for the podcast because I don't know if this season, but I hope soon I can make more in English. I would like, especially my
mm,
friends here, they're always telling me like I would like to understand your podcast, but it's in Spanish, so you should do it more in English. So yeah,
oh well, that's great. So you have
yeah.
a American audience as well.
Some of my friends they understand Spanish, or they actually use this podcast as like a Spanish class somehow. Oh yeah, love that. And I don't know. I always told them like you shouldn't use the podcast as your Spanish class.
Yeah, it's great. So
Well, yeah. Okay. So this is the first time that probably they will understand what is happening here. That's for real. How are you? How is it going?
oh, I'm doing fine, man. And a lot of work to do, a lot of writing down projects, and you know. Um, advertising my thesis, my thesis students, my undergraduate students, my graduate students. So this is very. I've been very busy for the last 2 months, and
Hmm.
I feel great because I have a lot of work to do, but at the same time, I'm tired, man. Combining my personal life, my children, all the research that I've been doing, gym. That I love the to work out and CrossFit and running and hiking and everything. So I feel sometimes, like very exhausted, but I'm happy, man, to
Yeah, I've seen you like busy doing all these conferences, and I mean you're pretty active now in social media like TikTok and Facebook. So this is pretty cool, man, because I know that you are more focused on like I don't know, like putting the science out of the lab and trying to get involved more people, especially people out of the research field. And this is
Mhm.
something that we are talking about in this podcast, right? Because sometimes, and we told this and we said this before in one of the podcasts, that sometimes research is good and the projects are good, but they don't leave the lab and they're just
Mhm.
looping and looping and looping in the lab for years and years and years until someone in the industry got the same idea and put it out in the world, right? So,
Yeah,
and this is important because scientists they have a lot of good ideas and good products and yeah, really good information that can help to improve a lot of things out of in the world. And sometimes we don't know and we don't have access to this information.
yeah. This is a true stuff. It's like sometimes we have we're scientists. We have a lot of ideas, and when the getting just one result give us the enough information to think in another and to study and to do different experimental things, and maybe study another field, and maybe to convert or to combine your field of expertise with other research area. And this is very important, but the thing is that many times we don't have the enough funding, and this is one of the obstacles that we have right now, mainly here in Mexico.
Right. Yeah, but this is something that also we said before, like here in the US, and we did our part of, well, you're part of your PhD, I did part of my master's, and some of the labs are sponsored by companies that they do investment in research to improve processing ingredients and products. And in Mexico, it's hard to get those sponsorships from
Mhm.
from the industry,
Yeah.
because private industry also, they have like their own department in the best scenario. Sometimes they don't, because the research is it's making a different country or a different facility, not in Mexico. Mexico is more like manufacturing thing,
Mhm.
and less like development. Sadly, sometimes that happens, and the universities could be this spot, or this place where these ideas can come to the industry, and sometimes the industry doesn't go to the universities for
Mhm.
this. It's
Yeah.
just lack of knowledge as well, because they sometimes they don't know exactly what is happening in the
What is
university, right?
happening? What are you doing? Uh-huh.
So sometimes like, oh, I don't mess with that because I don't know what they do. And then when they realize what people in the university or in these research centers are making, is like, oh, this is interesting. This can work. This can be fixing a lot of problems.
Yeah, no. Yeah,
Um,
yeah, yeah.
just yeah. So sometimes linking between industry and that,
The
like
academy
in the
and
academy is
research
hard
academies. Yeah,
in
it's sometimes it's hard.
Mhm.
And I think that one of the things that is between uh, us as scientists and population and industry is that we are very focused to publish our results, our projects, or maybe patents and protect
Yeah.
our ideas. But we are just focusing in one, field, for example, academy field. And we need to open and we need to express and to let it know the whole people, the whole population, what are we doing, what are we working with. And this is very important because population need to know and need to trust in scientists, because science and technology create new developments, new technologies that are making our life easier, so
Yep.
this is very important for us sometimes. And here in Mexico, Mexican people don't trust in scientists. This is very sad for us. So we are trying to break this obstacle for us.
Yep.
And this is why we are trying to get into the social media and report or talk about our results in not in a technical language. I
Right.
don't know if you, yeah, this is why different peers and different colleagues are trying to express the result, the projects, in a non-technical languages to letting know the people, the whole population, the development, the technological development that they are working with,
Yep.
for example. Yeah. Yeah.
And actually, well, this happened during the pandemic in 2020, because I think it was the first time in many years that the science was challenged to get results faster, and those
Yeah.
results like with good accuracy, and like and to be
Reliable, yeah, reliable.
accessible, yeah, and to be accessible with to everybody, right? And I mean, this is actually the main topic of this podcast is the that you have been working developing a new kind of vaccine for the SARS-CoV-2. I don't know that's the name in English. In Spanish, it is SARS-CoV-2. And the
COVID-19,
COVID, right?
the COVID-19, yeah.
So I would like to start because you're using a protein, a recombinant protein called CHIVAX. I mean,
CHIVAX.
CHIVAX 2.1. So, what is CHIVAX 2.1 and what is important?
Well, it's a recombinant protein that a colleague and his students created based on different proteins extracted from a COVID virus. So they created this protein. They assembled the whole unities, and they created this recombinant protein. So they applied or they administered this protein to different pigs, and the results showed that the 90% of pigs started to create protective antibodies against COVID-19. So this is the first finding that this recombinant protein really works against COVID-19. So they patent this protein. So the limit and the difficulties that this protein has is that they tried to administer or applied this protein through different routes, not conventional routes. For example, oral route or an intranasal route. So the main difficulties that this protein has is that the pH or the pH of the environment and the whole, for example, mucus or the physiological environment can degrade this protein. So they were focused on trying to investigate if the intranasal administration of this protein could be ideal or optimal to increase the immunological response, trying to immunizing the nasal mucosa. So their results were not good at that time. So they were trying to use different adjuvants that are conventional adjuvants that are very used in the pharmaceutical field. For example, aluminum. So aluminum on polysorbate 80, they are two main adjuvants that are used to combine with different antigens or proteins to increase the binding between the protein or the antigen and the nasal mucosa. So this is like a bridge, and they increase the binding or the absorption in the mucosa and increase the immunological or the immune response in nasal. But these conventional adjuvants, for example, aluminum or polysorbate 80, is that they can exert some adverse effects. For example, they can induce to irritation in the mucosa, in the nasal mucosa. They can increase the damage of the nasal epithelium, and they increase some allergic responses. So this is adverse effects could be seen like limitation of these conventional adjuvants. So Dr. Mosqueda, Juan Mosqueda, who is the leader of this group and the principal, the main brain behind the idea and the creation of this protein, alongside with Dr. Juliana Vega. They were talking to me, and they wanted to see and to investigate if some nanoparticles that I was working with could be a good adjuvants or good alternative adjuvants to this protein. So I proposed at that time to work with chitosan nanoparticles. Chitosan is a polysaccharide. It's a polymer, natural polymer that is obtained from chitin, and the chitin is the whole, like skin from shrimps and other crustaceans. And after this skin, after some chemical procedures, they can obtain the chitosan. So the main properties of chitosan is that it's a polymer that is highly biocompatible, and they can induce an immune response. It's immunogenic, so this is very important for us because they can create alongside the protein, they can create a synergistic immune response between the two components. And chitosan has this absorption of this adhesion to different tissues, including the mucosa. So this is these three properties were the main properties that we think that chitosan would be the optimal polymer, the optimal material to investigate the nasal administration of this system. I don't know if I respond your
No, actually, well, I have a lot of questions. I know this is like a first stage in this research. I know that the paper just was more like focused in the response, and like to try to prove that the vaccine doesn't have any allergic reaction
yeah
in the tissues, as you said, in the lungs and the nasal tissue. And because I have a lot of questions, so I don't know if we can go deeper or not with
and
this.
I don't know if I had the answers.
That's right, and that's science. You maybe don't have the answer, but it's like good because when you don't have an answer, then you have a opportunity
Opportunities
to
to
do research. Yeah,
yeah
right.
yeah to investigate
So
and try to answer.
yeah, so I mean the CHIVAX, it's well, for people just like to try and explain a little bit better, is like part of that try to uh, like be similar to part of the virus, and this part can react with the antibody in the subject, right?
Mhm.
So when you create this bounding between the antibody and the antigen, you start the immunological reaction in your body, and then just when
Mhm.
you start creating antibodies for that specific antigen, and you
Mhm.
fight the 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 illness, right?
First
So
is first is to teach to the body
right,
how to react
it's
and how
a
to
really
defend
long
itself.
process
Yeah, yeah, yeah.
because the cells, they try to improve the affinity between the antibody and the antigen, right? To make it
Mhm.
a lot, to do it super specific for that antigen, right?
Mhm.
And this process is cloning the cells in many, many, many times just to make this like super specific.
Mhm.
But this is triggered when a cell finds this antigen, right?
Mhm.
Uh, so the first question is, because the nose, it has a lot of barriers to protect the lungs and the respiratory system, right? So we have well, a lot of barriers, including the mucus.
The mucus.
Yeah. So when the antigen goes into the nose without protection, so I don't know if you already got this. Is like it activates an immunological response in the
Response.
subject?
It,
Because
it,
this is, it's different when it goes into the bloodstream, right? When it goes into the bloodstream, it's already there and interacting with your immunity system, right?
yeah. Well,
But
first we need to remember that, and we have a lot of barriers in our body, not that try to defend us against different pathogens and components that could be harmful for us, no.
mhm,
But for example, in the nose, in the nasal cavity, we have this mucosa. So the
yeah,
mucosa, underneath the mucosa, we have a lot of active immune cells that
mhm,
they are ready to combat or to fight different pathogens that interact with in the nasal cavity. So the immune response is all the time active. So this is very interesting because when you try to immunize an organism or a human being through the nasal cavity, the immune response is high. It's more effective.
mhm,
And if you compare with the conventional immunization, that is through an injection or through administration, intramuscular and in a intramuscular injection. So because there are a lot of different pathways that this antigen need to start and to trigger different activation that is humoral or cellular response. But in the nasal cavity, these immune cells are ready to fight it, to combat the whole pathogens and different xenobiotics that are present in the nasal cavity. So this is very important, and this is the first thing that we need to remember that is how the nasal cavity works. But we have a lot of washing mechanisms. For example,
mhm,
if you applied some or you administered some component in your cavity nose, the nose try to wash the whole components, like a protection reaction, right? The protect reaction, for example, and the whole components get out from the nasal cavity. This is one of the reaction that the nasal cavity has. And in the other hand, the protein or this CHIVAX that was created has very low adhesion or adhesion to the nasal mucosa. So if you applied this recombinant protein through the nasal cavity, maybe around the 10 or 9 10% of the whole protein can bind to the mucosa and start an immune response, an immune reaction. So 90% is degraded in the nasal cavity or wash it from the nasal cavity. So this is very important because in this scenario, the majority of the antigens not they are not able to increase or to create an efficient immune response through the nasal administration. So this is why we need some molecules or adjuvants that increase this binding or this absorption to the nasal mucosa. So when you are able to maintain or increase the time of the exposure in the nasal cavity, the antigens have the time to increase the immune response of the whole immune cells in the nasal cavity, and create efficient and a more efficient immune response. I don't know if I respond
Yeah,
your.
because this is important part, because we have a lot of spots in our body that they have like this immunity cells, right? And that's so if you go to those spots, you can promote this immunity response, right? So in the nose, this is something that happens. So it's a
Yeah.
good spot for this. But the other question is this, because you said that you are trying to promote the binding between the CHIVAX or
CHIVAX.
CHIVAX, right? Into the nose wall, right? And for this, we are nano-encapsulating this in chitosan to promote this binding. But and this is a question that actually I know that you're an expert of this, because designing the nanocapsule is really important on this part. So for this, uh, well, if you want to explain what are the different designs in the nanocapsules, because we have one that is covers and the center or the nucleus
in
part
the
of
in the in the core,
the
in the center
yeah
of the core.
the
Of the
core,
core. Uh-huh.
right? The core, it's like there's nothing there, and you can put the active in the core, right, and covered by the wall with
Mhm.
of the adjuvant. And other ones, the molecules are over the wall all around,
Mhm.
and so and so and so. There's like many ways to design this. So in this specific case, in this specific scenario, uh, how did you design the nanocapsule to carry the protein into the nose? And like I'm assuming you put it in the wall to have interaction with the nose,
Yeah, yeah, yeah.
but yeah, I don't know.
Well, this is a very good question, and I don't know how we did it. I'm just kidding.
I don't know how to.
No, well, first of all, we need to understand that this project is has or have two different objectives. The first one was to create the ideal or the optimal non-encapsulate system to incorporate the protein and maintain their chemical, physical, and biological integrity of the protein. This is very important. And the second was we tried to investigate if this system, this complete system, it was was safety to to administer in a living organism. This is very important because many scientists, or the majority scientists, wanted to create a micro or nano particulate system, and they go directly through to investigate if the system really works. But we changed the view, and we wanted to see if the system is safe to administer, because
Mhm.
maybe, for example, if I can demonstrate that the system is optimal to create, and then we can increase immunological response. But in the next step is to investigate the biosafety of the system. So many of these results can be challenging to investigate, and maybe the system is not safety to increase. So we wanted to investigate if the system was is safe to apply, because many systems that have been reported in the literature, they show it that when these micro or nano particulate systems are administered through the nasal cavity, they tend to increase some toxic effects, in the throat, in lungs, in liver. So we wanted to see in a preclinical preclinical size if this system that we have created, it could be safe to administer it. So this is to understand our project. So your question is really interesting, because when you try to create a particulate system, you need to see everything. The wall material, that is the material that will try to incorporate, will try to encapsulate different actives, and you need to think and to design an experimental condition and everything in the lab to maintain or to create the optimal physicochemical properties of this system, like such as size, surface electrical charge, morphology, but these conditions, for example, pH, concentration of reagents, and other variables, they can create the optimal system. But at the same time, when you choose a wall material, you need to see another properties, such as biocompatibility of the systems. This is very important. And other that I is the biodegradability of the system. This is very important, because if you applied in a living organism this system, and the system is not biodegradable, this is an issue very important in this area, because these non-biodegradable systems can accumulate in different tissues, in different organs, and with the time create different toxic effects or adverse effects, and we have seen it in other projects. So this is very important for us. So this is the first thing is to see the what material is biocompatible, biodegradable, and that is accepted to be used in this area. For example, in pharmaceutical area or biomedical area, need to be accepted to use in this area. And for other in the other hand is to understand that I'm going to try to incorporate a biological biocomponent or biological component that has a chemical structure, and this chemical structure exerts their biological effects. So if I have not the if I cannot care the whole environmental of the reaction, I can degrade the active. So this is a very challenge for us, for that scientists that we are working in the creation of different micro or nanoscale systems that we are working in the development of drug control release systems. This is very important for us, because when you try to incorporate or encapsulate an active, you need to take a look in the whole environment, the temperature, the pH, reagents, and other variables that to try to confirm or try to care or try to I don't know how to say it, to maintain, try to maintain the whole integrity of the component. So in this way, we investigate different materials. For example, some acrylate materials or other materials that can be used in the nasal cavity, and that we choose for different components and different properties the chitosan. So the next step was to investigate how we're able to incorporate the protein in the interior, on the inside of the particle, And this is very important because this was I think this is was your question. We can
Yeah.
create matricial systems, or we can create reservoir systems. Reservoir system is based on that in the core, in the cavity, on the center of the particle is located your active, and you can surround the active with a membrane of the different materials. But we can create as well, depending of the experimental condition and everything, a matricial system, which indicates, which is based on the active is dispersed in the whole system. You can find it in the center. You can find it very near to the surface, or even in the surface created a crown. It's called in the crown. It's like um, monolayer that is surrounding, or this active monolayer is surrounding the whole system. This is a matricial system. So we after different conditions and after different experimental assays, we found that the ideal experimental condition that maintain the chemical and biological structure of the CHIVAX was creating the system, the whole system, using some experimental designing to create a matricial system. So this was the first result that we obtained, that we got, and we were able to maintain after this synthesis, this development procedures. We were able to maintain the whole structure, chemical structure, and the biological effects of the protein. I don't know if I
Yeah.
were
So
able to
did you choose, so the nanocapsule is more designed with having the protein in the core, right, surrounded by the chitosan?
Yeah.
Right?
So we
So
created this matricial, and we created a matricial system, which indicates that the active or the protein were located in the center, near to the surface, or even in the surface, creating this active monolayer surrounding the particle.
right. Okay. So the proteins will be all over the
All over.
nanocapsule.
Yeah.
Okay. Gotcha. And I mean, in this case, I mean, the protein that is on the surface, I think that has more interaction. But you mentioned it's something interesting because this is this degradation of the nanocapsule. So I guess that the chitosan, did you work with chitosan before, and you know more or less what is the the time, the frame of time that it takes to degrade the chitosan in the body? So is this like a slow releasing thing, or because I guess also immunological system works by probability, right? So you have to get that antigen like enough time in the body to promote this immune, this immune response.
Okay.
So I don't know if that was another thing that you put on the table when you designed the capsule.
Well, based or focused on the chitosan, the biodegradation, the time, the biodegradation time, or the biodegradation profile, it depends of the molecular weight of the chitosan, the concentration of the chitosan that you are using to create the system, the viscosity of the solution, the precursor solution from of chitosan, and everything. So this is very important because depending on the concentration, the molecular weight, and the time, and the concentration of the triple phosphate, which is the the cross linker.
Hmm.
I was forgetting the word in English. It's the cross linker. Depending on the concentration of the cross linker, you can create a different system with different average sizes. So this is very important because the system that has larger particles, they tend to degrade in with increased time or with more time in more time. So this is
Hmm.
very important. So we investigated in this case for the development of the system different concentration of chitosans, and we observed that the more chitosan concentration, the larger particles that we obtained. So this is very important. So and we studied the binding of the mucin. The mucin is the main component of the nasal mucosa, and we observed that the smaller particles have a larger or increased absorption of the mucin. So we observed that. So with the increased concentration, with higher concentration of chitosan, we created the larger particles. So the more concentration, the larger particles. But the more mucin binding was found in the smaller particles. So that was we choose to work only with the smaller particles, which were built or were developed with the lowest concentration of chitosan. So after that, we investigated in a simulate nasal fluids the release of the protein, of the recombinant protein, and we observed that we had an immediate
Hmm.
or burst released. For example, in the first 2 hours, we release around the 60 or 70% of the protein, and it depends because sometimes some scientists or some experts of the vaccines field, they claimed that you need to create a systems that release the antigens in more time, because the more time indicates that the active has more time to interact with the mucosa and increase the immune response. But on the other hand, other scientists claim that an immediate release or burst release could be beneficial to increase or to generate, in this case, an immune response. So there are two different point of views between the experts, but with the system that we created, we observed that in the first 2 hours we release around the 60, 70, or 80%, around 80% of the protein, and we calculate using some mathematical methods, and we were able to hypothesize that the release were the release phenomena of the protein were based on the swelling of the particles and then the degradation of the particles. So the pH of the simulate nasal fluids that were around that was around 6.8, I think. These pH conditions increase the biodegradation of the particle that we created. So I think the degradation of the particles of the system that you create using chitosan, it would change depending on the concentration of the molecular weight, and even in the part of the body that you administer the system. So in the case of the nasal cavity, the pH, the presence of the mucosa increase the degradation of the system and release in the two first hours the release of the protein, and this could be beneficial. So we need to investigate. The next step would be to investigate if this release profiles could be good to enhance or to improve the immune response in a living organism. I don't know if I could explain.
No, yeah, and this is important because all this part of designing, it's the main core and doing all this like um, simulations, and because this is not all this work goes out of the real living body.
Yeah.
This not you could be an animal or human, but and yeah, so all this designing, how the protein is released, how this interacts with the fluids in the nose, and all those things, yeah, are really important. But the part, like the big question is like, is this triggering immune response? So I think that you work on this also in the paper. So did you get any immune response in this animal model that you test?
Okay,
Tested?
this is a really good question because we have not reported this part. We were
Oh, yet? Sorry.
no, not yet, not yet. We were able only to investigate some immune
Don't say
response.
anything then.
No, we were able to investigate a tiny immune response, and we observed that the system are able to increase the immune response in a 10%, around in a 10%. This is not a significant increase if you compare with the controls, but it was telling us that the system really works.
Right.
But we were not able to continue these experiments because we were lack of funding,
Found
like
it.
funding, we
Donate here. This is
yeah,
the
please fund us, and we need
this is the bank account is going to be appearing
yeah,
here in the
please
banner.
help us to investigate this part. But
No, I'm asking this because I know that in the paper it says that you test if the capsules they create any like allergic reaction, and I think this is the main result in the paper, that you got like tissue, like I don't know, like different tissues from nose, lungs, and everything in the
okay,
rat.
get it, get it, get
Yeah,
it.
yeah, yeah, yeah. But I didn't see, and this is because but I was reading and I thought like, oh well, this is fine because you know first you have to prove that this is not causing any allergic for anybody. I mean,
Yeah,
you explained at the beginning of the podcast that this is one
yeah,
of the things, but then is the immune response as
yeah.
well, because I don't know, there's a lot of questions between both results.
Yeah, no, I know, but we were focused to investigate if the system was safe to apply. So after we administered to the animals, these experimental animals, I don't are you
It was rats.
okay?
Rats. I'm
We
okay.
were using rats, and after we administered the
I
systems, we wanted to see if the system was totally safe to administer, to apply. So we investigate, and after we sacrifice the animals, we investigate different organs, for example, lungs, liver. That are organs that are really connect or really correlate.
fine.
Okay, I'm back, man. So we wanted to investigate the possible adverse effects, in nose cavity, and the whole up the respiratory system, the upper respiratory system that includes lungs, turbinates, and everything. And as well, we investigated if this system could affect liver, lungs, kidneys. And we observed that the presence or the administration of our system did not affect or did not modulate, do not change the histological and well, the main focus of the project was to investigate if this system was safe to apply. So we focused to investigate if different organs that are related to the immune response, such as upper respiratory system, that includes turbinates, lungs, and the liver and kidneys as well. We investigated if after the administration of our system, these uh, these organs have changes or uh, some changes in the histological level that could tell us that our system is not safe. So we observed that after the administration of our system, the organs did not show any adverse effects or toxic effects. So with these studies and with these histological assays, we could confirm that our system is safe to apply. So the next step is to investigate if our system that now we know that the system is optimal in design. The protein maintain the chemical and the biological properties, and after in vivo administration, the system is safe to apply. So the next step is to study if our system could increase or could trigger efficient immune response using the immunization of the nasal mucosa. So this could be the next step, but we need funding to do that. So was my second in vivo study, in vivo model study, and that this one and this project was the most expensive investigation. It was around, I don't know, like, well, for here, for Mexico, it's very expensive. It would cost around between 20 or $30,000 in total. So this is very expensive because the animals, experimental animals, are very expensive. The reagents, the maintenance of the experiment animals are very expensive. The study during the in vivo protocol, during the administration protocol, and after we sacrificed the experimental animals, you need to obtain and recover the whole organs and maintain it in different storage conditions to maintain their histological that the histological level,
to keep
and
the proteins,
everything. So this
everything
is very
necessary
expensive because how we need ultra frozen, we need to ultra frozen equipments and reagents to maintain their structure, the biological components, everything. And after that, we need to study the whole components using other reagents or other biological kits, everything. And this is very, very expensive. So we were, well, we right now we are very happy with the results of the project, but we are we are looking forward to obtain some funding to continue this study. So this is very important for us because the other result that we have not showing in this article, in this manuscript, is that we observed that after we incorporate the protein inside the particles, we maintained during 10 days the immunological response of the system, which means that you're not, for example, one of the problems in vaccines, if you take it out, the vaccine from ultra frozen, for example, that has storage around the vaccine need to be stored around minus 70 Celsius degrees. That I don't know if you compare to Fahrenheit.
I'm
To
still don't
Fahrenheit,
do it.
like minus 10 Fahrenheit degrees. I don't know.
Right.
And after that, you need to apply the vaccines. If you are not able to apply the whole unity, the whole vaccines, they are not able to come back to the storage, and so the vaccine are degrading after you take it out from the frozen storage. So with this system, we are able to maintain the protein using a conventional freezer, for example. That we are able to maintain during the 10 days the chemical structure of the protein and the biological effects of the protein during 10 days, which means that you are able to take it out from the conventional fridge. You go into different communities of population. You administer the vaccines, and the vaccine that are not able to be applied, you can come back to your house, to your lab, and storage again the system, and you can repeat this cycle around 5 days. So this is very important for us, because with this kind of system we could change the vaccine protocols of a country, for a specific country. We were thinking here in Mexico, and we could be helpful to I don't know how to say that. It's like we are thinking that we are able to decrease the economical losses in a country that are related to whole vaccines that are degrading because they were not able to be applied. I don't know.
Right. No, yeah, because this happened in the past when the COVID, after the second round of doses or the third round of doses, many people didn't go, and they were pretty much like giving for free the vaccine, because as you said, once that is out of the fridge, it cannot come back and it starts to degrade and everything. So this is important because the context, the social context in Mexico is different. And as you said, there's a lot of communities that they don't have even electricity or whatever, and just to keep safe the vaccine in a cooler is fine, right? So yeah, so yeah, it's very interesting. I think the next steps are pretty challenging, because these clinical protocols, there are always like a lot of steps, and you have to get a lot of information, and from here to proved in humans is really long way, as you said, an expensive way as well.
And this these results help us to create or give us the idea to create other system, a microparticulate system, and we are able to incorporate different bacteria, and we are able to store it to a different time through time. These bacteria are able to maintain their biological properties and everything using a conventional fridge. So this is very important because we are helping to increase to maintain the biological properties, effects of different bacteria that could be beneficial for different fields, and we are trying to help some laboratories that has not the economical support or the economical way to obtain this ultra freezer. I don't know how to say it. This, so they are very,
I don't know the name in English as well. So.
It's like ultra. Oh, I forgot the name in Spanish even.
Ultra congelador, right?
Ultra congelador. Entonces, so this is very important for us because this project gave us the idea to create other systems using other natural polymers. We are using today alginate. So this is a food grade alginate. So right now we are creating these storage, very low cost storage systems to different bio components or even bacteria, and you can storage in using just a conventional fridge around 4 and 10 Celsius degrees. So this is one of the extra beneficial that gave us, or that that gave us this kind of results.
Right.
Very low cost storage system
Yeah.
based on natural polymers.
Yeah, that those are ingredients that were really used in food industry and
Mhm.
really accessible for, like most of the industry, I guess, and most of the
Mhm.
academy. Sorry. Yeah.
Yeah.
Well, yeah. All right. Well, thank you for coming to the podcast. This was a new experience. I have
Yeah.
this podcast in English.
Yeah.
So I felt like I haven't talked about science in English since master's degree. So that was like 20 years ago. So it was really interesting to do it again. But
Yeah.
yeah, thank you. And I hope the next time you can bring us like new results in the research. I know that you're working a lot of things right now. So this is just one part, branch
Yeah.
of your
Right
research.
now we
Yeah.
are into the entrepreneur, scientific
Yeah.
entrepreneur. We are trying to sell to different investors and enterprises our technological developments based on
Yeah.
nanotechnology systems, and that we are working in agricultural fields and biomedical fields. We are trying to sell and to obtain, some funding, or some I don't know how to say
Investor.
it, investors to, to protect the idea and then sell
Mm.
it. Yeah,
To get the patent. Well, there's many different ways. I think the
yeah,
patent is the less good one of those. But yeah.
yeah,
But anyways, well, thank you. And I don't know, do you want to give us your social media where people can follow you? And
yeah.
because you do
This-
a lot of scientific divulgation. I don't know, that's a
uh,
word that I don't know in English, but yeah, for sure, maybe it's that one.
okay.
Uh,
Okay, this is my Instagram. Is Héctor Pool 40. You can find me in Instagram and TikTok. It's free to watch all my my
you do a
TikTok
lot of
videos.
videos, yeah.
Yeah, I have a lot of videos that I posted in during my workouts in gym, and even my research work. So this is in TikTok. You can find me as Hector Pulfori, and then this is my Facebook page, or my Facebook profile is Hector Reyes Pool. So because my first last name is Reyes and my second last name is Pool,
Cool.
you need to know that we here in Mexico we use two
Two last
two
names.
last names. So, but almost the majority of people know me just as Hector Pool. So because my second last name is not that common here in Mexico.
Dr. Pool.
Doctor Pool.
Anyways, well, thank you for coming. Yeah, and people where to find us, follow us in Spotify and YouTube as Just Gente Podcast, and hopefully I will have you soon here.
Yeah,
Thank
it
you,
will be
and see you next time. Thank you.
in the time. Thank you.